I'm Rinako Nakagawa. I'm here to present the work I performed in Elena Vigorito's lab as a Marie Curie Incoming Fellow at the Beverham Institute in the UK.
I'm Elena Vigorito. When I was group leader at the Beverham Institute, my lab was interested in understanding the molecular mechanisms that controls B lymphocyte activation, which underpins effective immunological responses. We previously discovered that the oncogenic microRNA 155 is a key player in the antibody response in a B-cell intrinsic manner. Here we uncover the cellular molecular basis for this process. Over the course of an infection, B cells produce antibodies with increased affinity to invading antigens. This is achieved by the coordination of three main processes. First, expansion of antigen-specific B cells. Second, acquisition of random mutations in the antibody binding sites. And third, antigen-driven selection of B cells expressing antibodies of the highest affinities. These processes occur in structures called germinal centers within secondary lymphoid organs. Germinal centers are composed of two main regions, the dark zone, where B cells actively proliferate, and the light zone, where B cells are positively selected.
To identify germinal center B cells expressing MIA-155, we combined a MIA-155 LAC-Z reporter system with a B-cell receptor transgenic model that recognizes hen egg lysozyme. Then we adaptively transferred health-specific B cells carrying a MIA-155 reporter allele to mimic a physiological immune response. At the peak of the immune response, we single-cell sorted GC B cells as dark zone or light zone B cells that were further separated by LAC-Z status, and MIA-155 expression was measured. LAC-Z positive cells express higher levels of MIA-155 than the LAC-Z negative ones, and MIA-155 is mainly expressed at the highest levels in a fraction of light zone B cells. Given that the proto-oncogene c-Myc is expressed in the subset of antigen-selected light zone B cells and is critical for GC maintenance, we tested whether the two molecules are functionally linked. First, we assessed MIA-155 and c-Myc co-expression by using a c-Myc GFP reporter mouse. Upon adaptive transfer of health-specific c-Myc GFP B cells and HEL immunization, GC B cells were sorted, and we measured MIA-155 expression. Light zone c-Myc positive cells expressed the highest levels of MIA-155, demonstrating that a subset of light zone B cells co-express c-Myc and MIA-155. As c-Myc has a pivotal function in growth control and apoptosis, we hypothesized that MIA-155 may mitigate apoptotic effects driven by c-Myc. We used our adaptive transfer method with health-specific c-Myc GFP cells in MIA-155 sufficient and deficient backgrounds and measured apoptosis. Light zone c-Myc positive cells were the most susceptible to apoptosis in the absence of MIA-155, suggesting that MIA-155 provides survival signals to c-Myc positively selected B cells. Next, we wanted to address how MIA-155 regulates survival. JARID2 is a pro-apoptotic molecule highly expressed in MIA-155 deficient light zone B cells and is directly inhibited by MIA-155. We tested whether increasing the expression of JARID2 in GC B cells would mimic the effect of MIA-155 deficiency by ectopic expression of JARID2 into activated B cells, followed by adaptive transfer. This led to increased apoptosis in light zone B cells, suggesting that MIA-155 inhibits JARID2, allowing the survival of positively selected light zone B cells. We have shown that MIA-155 cooperates with c-Myc, allowing the selection of high affinity B cells in the germinal center. The concept of two cooperating oncogenes in B cell selection is important not only for understanding the physiology of antibody responses, but also to understand B cell malignancy.
Expression of c-Myc and MIR-155 is increased in some lymphomas. Understanding the molecular interaction between these two oncogenes may shed some light into their pathogenesis or may assist in potential treatments.